Oh, you've spilled the beetles.
Oh shit, no.
Oh, the beetles.
Don't worry, look how we take the situation.
Oh my gosh, look at them. Look at these guys. What? Hey guys, I'm super excited to be here today with my friend Margherita, who is a biologist here at Cambridge University, and today we're going to be doing a behind-the-scenes look at what real biology research looks like. So welcome Margherita, it's lovely to have you here. How are you doing?
I'm doing well.
That's
Thanks.
good.
Welcome to the Zoology Department. I'm very happy to have you here.
Thank you very much as well.
So I am a third-year PhD student in developmental biology. And that means that I look at the development of embryos, and in particular I work with insect embryos. So today I'm gonna walk you through some experiments, and hopefully all the experiments will work.
Oh, definitely they're gonna work.
Yeah,
I have
definitely.
full trust in you.
And if they all work, we should be able to see some live embryos glowing in the dark with some fluorescent proteins.
That sounds amazing.
Yes.
That sounds amazing. I'm looking forward to it.
Me
All
too.
Right. Let's do it then. Let's go.
Okay, so we're going this way. The zoology department is all around us. Where I work, in fact, it's not the old building, but it's a new building, and it's called the David Attenborough building.
Big man himself.
Those lights, that's where the lobby is, and that's where we're going.
Okay, so you got your lab coat on now, so you're a proper scientist.
I am.
Everybody knows if you don't have the lab coat on, you're not doing science.
Absolutely. That's what
Absolutely.
makes me a scientist.
What is the first thing that we will be doing?
The first thing is I'll show you where the beetles live, and I'm gonna show you how we can get, through sieving, the embryos out of the flower and separate them from the adults that are laying the embryos.
Okay.
And then we're gonna prepare these embryos for injection, and then we're gonna inject them with some RNA. So hopefully we should see their nuclei and their membranes glowing.
That sounds pretty cool actually. You're gonna make some beetle babies glow,
Yes.
glow in the dark.
Yes.
Okay.
That's the plan.
Well, without further ado, shall we
Yes.
let's see some beetle babies.
Okay. So this is the Beetle Incubator. These are all my beetles. You see them? They live in flower.
Oh, they're just little beetles.
They are little. So these are the sieves. So this is the smallest one to separate the embryos from the flower, and this is the big one to separate the adults from the flower.
So we've got the flower.
Yes.
And the beetles lay their eggs within that flower?
Yeah.
Okay, and then it's just a matter of separating the rest of the flower, the beetles, from their eggs.
Exactly.
Okay, this looks a lot like baking. Not that I...
You know, it's all about baking. So that's what my first supervisor ever asked me when I said I want to join his lab. It was like, do you like cooking? Do you like baking? Because it's all about that. It's the same thing.
Oh, you've spilled the beetles!
Oh shit, no.
Oh, the beetles!
Don't worry, look how we take the situation.
Oh my gosh, look at them. Look at these guys. What?
I know, it's ridiculous. There we go.
Okay, we...
This is the beetles. It's full of beetles. So after the sieving, now we have just plain flour here. We have all the adults here. And then these are our embryos, these small white dots. We need to treat the embryos a bit before we inject them. The embryos are surrounded by a membrane, which is hard, and therefore the needle for the injection cannot get inside the embryo if the embryo retains this membrane. So what we're gonna do is use bleach, and we're just gonna wash them very gently in this solution so that the outside membrane will dissolve and they will still remain alive and completely fine, but we can inject them after that.
Mm-hmm. Okay, so they'll still be alive, unlike those beetles in the vacuum cleaner.
Yeah. We bleach the embryo, like my hair, so now we can inject them. But to inject them, and this is ironically the most time-consuming bit of this procedure, we need to align them. And so it's easier when we will go to the microscope with the needle to inject them one after another if they're all in a line. But this will take a bit.
So the next step is where stuff gets crazy,
Yeah.
and you literally stab these embryos with a massive needle,
Yeah.
which is like as big as the embryos themselves.
Yeah.
You stab them, and then you're injecting this special cocktail of genetic material, is that right?
Yes, it's
Into
RNA.
the RNA. And what does that do to the embryo?
So the RNA is a copy from a fragment of DNA. The RNA we are injecting is a copy of some genes, okay? And the embryos can recognize this RNA and translate it to make proteins out of it. So we give the embryos just the RNA, and if the embryos survive the trauma of the injection, they will translate this RNA into proteins, so thanks to their cellular machinery. So the embryos itself does most of the job.
Okay.
We just give the raw material, and the embryo will process this RNA and make some fluorescent proteins.
So you're injecting this RNA thing, and then the cell recognizes that RNA, and that's basically RNA is the encoding for different types of proteins.
Yeah.
Is that the...
To make it very simple, yes. This kind of RNA, that's what it does.
Okay.
It's called messenger RNA. So
Right.
it's a little copy of a fragment of DNA that then gets transported outside of the nucleus, because the DNA lives in the nucleus.
Yeah.
So you make a little copy with the RNA, transport it outside of the nucleus in the cytoplasm of the cell, and that's where there is all these different proteins, and there is the cellular machinery that will make the protein.
And it just automatically recognizes this RNA, like
Yes.
even though that genetic material isn't from beetles, it's from some other animal or organism, I guess.
I did it in vitro, so I made it myself. But the cool thing with all living things is that we all come from a common ancestor. So we all use DNA, and my DNA is the same DNA that you can find in a frog or in a bacteria or in an algae or whatever I can make in vitro myself. So that's the cool thing. We can just use sequences of DNA from other animals and put them in different animals, and they will still work.
That's so cool though. That's that's full on science fiction. It's the sort of stuff that you see in like some bad superhero movie, and you think, okay, this is complete, bullshit. Like this could not but that's literally what you do. You just inject some like genetic material from another animal, and it actually changes the embryo, and well, in this case, makes it glow in the dark.
Yeah.
Okay.
Don't hang up. Can we be friends again? I give up.
A lot of time has passed now. The beetle embryos have had the opportunity to make these green proteins, these red sort of fluorescent molecules, and now it's time for the microscopy part.
Yeah.
So just very briefly, what's going on in this experiment with the microscopy?
Yeah, so the beetles, as you said, now have made these fluorescent proteins. If you just look at them with your naked eye, you wouldn't see these fluorescent proteins, obviously.
Yeah.
Because the way fluorescent proteins work is that they need to be excited by the laser. They get the energy, right? The electrons jump to an excited state, and then when they jump back to their normal state, they will emit a photon in a specific wavelength. So we go to the microscope and we use some lasers to excite these fluorescent proteins for us to be able to see them. Okay, so they look fine.
Okay.
I'm just searching for one good embryo. There we go. You see that?
Ooh, okay.
So these are the membranes. Okay, it worked. So this is our embryo. And this is the green glowing membranes. So it's this fluorophore we're seeing is neon green and is attached to this GAP43 protein. And it basically just makes the membrane glowing. So we injected the RNA, the embryo took the RNA, liked it, thankfully, and made some proteins with it. And this protein is a protein that goes in the membranes and is attached to our little fluorophores that we can excite with the laser. And we're going up and down here. So this is the top of the embryo.
I mean, that is just super, super cool that you can even do this, right? Like, it's awesome.
And we can go deep in the embryo. See different layers inside. And then the other one, obviously, is the nuclei.
What is the difference between this one, the red color, and then the green color?
So the red color is histone, it's called the protein. Basically, the DNA is wrapped around these proteins. So we couple this protein with the little fluorophores, and therefore this protein will localize in the nuclei, and when we excite it with the right wavelength, it will give us red nuclei. And the other one is the same thing, but instead of being a histone protein, is a membrane protein. So when we have both of them, they look like that.
Oh, that is super, super cool.
And this is live. Like, this embryo is alive.
So
completely alive.
This would grow into an actual beetle.
Yes, and what we will do now is to set up a scan, and we can have a time-lapse video. So we'll take a scan every 5 minutes or something, and then what we will see is the embryo moving and the membranes and the nuclei moving.
Oh, that's so cool.
Yes.
We'll essentially see what happens to the embryo as it grows over time and as it develops into a beetle.
Yes.
The biggest question I think of all is why study these embryos? You know, what is the main point of studying these transformations that happen in these beetle embryos, and what can that really teach you about biology in general?
Yeah. So obviously I'm interested in the development of insects for its own sake, because I find development in itself something stunningly beautiful. The fact that we all come from one cell.
Yeah. However, is there a bigger picture as well?
Yes. So for this, we need to delve into my research question. The main process that I look at is called EMT. This is a cellular transition from an epithelial to a mesenchymal state. An epithelium is a sheet of cells that are all more or less connected. An epithelium could be something like a skin, and is what is around all your organs. So it's a barrier of cells that are tightly connected and linked to each other. Obviously, there is a second bit, which is mesenchymal, and this is a transition. And that's also is the reason why I look at animals live, because cells that are alive, sometimes they decide that they don't want to be in an epithelium anymore, and they want to transition towards a different state called mesenchymal. So mesenchymal cell, and this is obviously very generalized, but there are cells that are not really connected to each other. They're loosely...
Like a free-floating cell that isn't part of the structure.
Yes. So the reason why this transition is important is because it's ubiquitous in both development, physiological, and pathological conditions. So the main interest people have in EMT is because it is what causes cancers to undergo metastasis. So a lot of cancers are epithelial cancers, and they're called carcinoma. And it means that all the cells are tightly connected somewhere, which means those cells will stay there. Some of these cells will get triggered by some factors to transform themselves and become migratory. So when a cancer does that, it means that your cancer cells will enter your bloodstream, go around your body, and then undergo the reverse transition, which is called EMT, and form a new cancer somewhere else in your body.
And that's
very bad. EMT as I was saying before, is not just found in cancer, but is found in other physiological and developmental processes. So for example, if you have a wound, the cells around it will undergo the transition and crawl and close it.
Right, okay.
And also during development, when you have to organize different cells and you have some cells have to migrate somewhere else and form different organs or different regions in the embryos, some cells will become to pass from an epithelial state to a mesenchymal state and move around and create new tissues. Yeah, this is a Drosophila embryo, and if we cut it here and if we look at it from the side, it will look something like that. So all the cells are on the surface.
So this is an embryo in the very, very early stages of going from a single cell to multi-cells and becoming eventually the proper animal.
Exactly. What happens is that all the cells are on the outside of the embryo and they need to organize themselves to create new tissues. And so what happens is that there is a folding event in which these epithelial will basically fold inside and do something like this. And these cells are still tightly connected to each other because they're still an epithelium, right? And then these specific cells that went inside, they will undergo EMT. So the outside remain an epithelium and inside the cell will be mesenchymal.
So essentially what you're saying is that in the very early development of these embryos, you always have this particular EMT transition. So from cells on the outside of the embryo,
that they go inside.
And becoming more free form, being able to kind of do their own thing. Yeah, so these embryos give you a very controlled and precise way of studying this process and learning general concepts about how the biology works. Just before we end, I just want to ask you one last question. So for you, what was the main motivator for doing a PhD? Why did you want to do this at Cambridge, you know?
I really found my place when during my master's studies, I started doing research. So I really loved doing my master's thesis and working in a lab. I like the environment. I like the fact that you're working alone, but you're also working with other people. There is a lot of collaborative efforts that go into science, at least that goes into developmental biology. And the fact that also, on one side, is good that you don't have really a routine, but on the other hand, it's also bad. But if we focus on the good part, you are free to do whatever you want. Like every day you can wake up and come up with something original, new, and that nobody has done before. And nothing or nobody can take that away from you when you do research. Like no matter what research you're doing something new. And that's incredibly exciting. Like that's the most exciting thing I can imagine. And that's pure research. Like this is why I love doing research. It's just I'm contributing at some level to humanity knowledge, you know? I'm contributing to our collective knowledge, and that is beautiful. Like I'm exploring mysteries that nobody has explored before. And it's not that, and this is fundamental in science, I'm not gonna keep what I found to myself. It's exactly the opposite. As soon as I find something, I wanna scream it to everybody and make it public, right? And at the end, there are some names, there are some scientists that become famous, but most scientists won't become famous, except in a specific field. But still, what you have done will remain in some sort of way, and will hopefully help other people to find other things. So you're building up this collective knowledge, and that's quite exciting.
100%, like that is a beautiful sentiment. I totally agree.
I'm keeping these all positive, honestly. There's all this shit about academia and publishing, but we're not going there, right?
No, we're just projecting good vibes, okay?
Good vibes. Come do research. I think another thing I would like to say to somebody that is considering a career in research and especially in biology is to never forget what biology is really about, and it's about life. And so go to museums, go see animals. If you see an insect on the street, like stop, watch it. It's
Don't do to
beautiful.
the insects what we did to the beetles.
No, but you know, like be.
But yeah,
you have to be amazed by the wonderful
Yeah.
nature of life.
100%.
Like life is incredible, and we should always cherish it in every form and shape.
Yeah.
And so, if you want to be a good, passionate biologist, never forget why you're researching what you're doing, and you know, be passionate about life, and be passionate about animals, and be passionate about all these things.
That's awesome.
Yeah.
Yeah.
Good vibes.
That's good vibes, guys. Hey guys, so that was my behind the scenes video of biology research at Cambridge University. I know it was a bit of a long one, but for me at least there was just so much awesome science that I couldn't leave out. And in fact we have a lot of extra footage on these genetically engineered fruit flies which I may make into another video at some point. I have to say, massive thanks goes out to Margherita. As you can see, she's genuinely a really inspiring person and an excellent scientist, so it really was a huge amount of fun making the video with her. As always, if you want to see more amazing science, subscribe to the channel. If you liked the video, give it a like. Even if you thought it was okay-ish, you could still give it a like. And if there's any particular type of science that you would like to see more of, let me know in the comments and I will do my best to make that video for you. Cheers.